Hey everybody, thanks so much for joining us from Genes in Space. My name is Mark Bliss. The Genes in Space competition has asked for your questions, and we're here to answer them. So joining us are our Genes in Space experts that are here to provide some guidance. Today, our guest is Pristine Onuoha. Pristine is the winner of the 2022 Genes in Space contest. Her experiment, studying the mechanisms of telomere lengthening observed in astronauts, launched in June of 2023. She's a 19-year-old college student at the University of North Carolina at Chapel Hill, and she's working towards a career in biomedical research. She enjoys the process of discovery and is particularly passionate about genetics research related to aging and cancer. Welcome, Pristine.
Hi, Mark. Thanks for introducing me.
Absolutely. We got a whole bunch of questions from students, from teachers, from folks that are just interested in the program. Our first question is, what is Genes in Space, and what do you think are the benefits for students that are participating?
Genes in Space is a national contest largely sponsored by Boeing and the Mini PCR biotech company. And it's a program where students in 7th and 12th grade can submit a hypothetical genetics experiment investigating a topic related to space travel. The winner of the contest gets their experiment developed and conducted on the International Space Station by astronauts. But for all participants, I think Genes in Space helps you think like a real scientist, which is invaluable for investigating the pressing scientific questions of our age. Plus, Genes in Space is just a really unique opportunity for young students to explore the field of space biology, which is an incredible emerging field that can teach you so much about science.
All right, so our next question is, how can one find out about the problems and topics that are prevalent in space biology and research? What are some of those topics? What advice do you have for them?
When I was originally starting to participate in the Genes in Space contest, I didn't really have much of a background in space biology. I didn't really know what it was or what research in that area could look like. But what I didn't know encouraged me to just learn more. So when I was trying to learn more about space biology, the first thing I did was just look to the internet. There are so many studies online related to space biology, like the NASA Twin study. And in addition, NASA has a lot of pages that discuss the effects of spaceflight on astronauts and the unknowns surrounding those processes. These effects include altered blood circulation, altered genetics, and an altered immune system. So I think these resources are a good place to start off with for inspiration about what you could write a Genes in Space proposal on.
Yeah, there's also a ton of resources on the Genes in Space website. We'll make sure that we post our resources page below. All right. Another question is, my students don't want to enter because they're intimidated by the past winners. Christine, should they be intimidated by you? How can they encourage these students to apply?
You don't have to be someone with a really developed background in space biology or STEM to participate in Genes in Space. It's open to any student who's just really passionate about exploring the process of research. And so I think just what's most important is that you're willing to learn throughout your experience. From my own personal experience, coming into a challenge, I hadn't had much of an experience with space biology, but I was really encouraged to learn about it so I could develop a strong research idea. So as long as you have that determination to really explore, I think that really propel you.
All right, Christine, besides winning and going to the launch, which I know was a huge event, I was lucky enough to be there as well. What would you say is the best part of Genes in Space? What have you unexpectedly learned from this experience?
For my entire Genes in Space experience so far, I gained a better appreciation for having flexibility. There are many steps in the timeline of my experiment when I needed to adjust and adapt to new concerns. When I progressed to being a semifinalist in the competition, I learned that my proposed experiment, which had involved the genetic analysis of astronaut white blood cells, would need to operate in a different way without human samples. And so, with this new challenge, I worked to rework my experiment in a way that still stayed true to my research goal of investigating how telomeres change in space. Most of all, even after winning the competition, my experiment changed in order to address an unfilled aspect of the ISS toolkit. So, going through this process of experimental design and redesign really helped me understand how research is just a reiterative process and that roadblocks are really opportunities for redirection in a more positive direction. I'd also really enjoyed getting to learn and grow with my Genes in Space mentor, Anna Carla. She provided me with a lot of guidance regarding the science and presentation of my experiment. But in addition to this, she was just someone who I saw as a really supportive figure and who really believed in me. Also, genes in space unexpectedly spurred my passion for space biology, and now it's a field I really want to pursue in the future. I hadn't known much about the field beforehand, but as I was writing my proposal, I learned just how interesting and unique it is.
Pristine, you were just talking about being flexible and learning to be flexible. How far was your initial idea from your final experiment in your genes in space application?
Yeah, so my original proposal involved detecting the various levels of bacteria in the gut microbiome of astronauts. I had written an entire proposal based on this idea. However, before hitting submit, I felt the proposal I had written wasn't really specific or as compelling as it could be. It described an experiment that wasn't really simple and wouldn't yield results that would be super relevant. But this realization just encouraged me to go back to the drafting process to write a proposal that I felt tackled a unique problem in an interesting yet fairly simple way. And this final proposal I submitted was on a completely different subject, the link changes of astronaut telomeres. Given this, I would encourage you guys to keep an open mind during your proposal writing process because you never know what idea might really end up captivating you.
All right, Pristine, we just have one last question for you. Obviously, you had a winning experiment. What advice could you give students who are about to pick a topic for their own genes and space application?
Yeah, so I would encourage you all to design experiments that are specific, detailed, yet simple, and make the most of the Genes in Space toolkit. Having a specific experiment means that your idea addresses a question that isn't too broad. It also means that your experiment would lead to results that are super relevant to helping us address your research question. For my proposal, I was interested in investigating how astronaut telomeres change in space. But since this is a really broad topic, I did more research to learn about any specific gaps in knowledge we have about that process. For example, whether it's harmful, whether radiation causes it, or if something like stem cells drive this change. I ended up choosing the stem cells idea because I thought it would be a really compelling way to pursue my broader research question. But I definitely could have went with any of those other research ideas. So with my research question, my next step was to design an experiment to tackle it. Given how the space environment lacks a lot of resources, it's really important to design an experiment that's resourceful and fairly simple. With my proposed experiment, I wanted to see whether there were more or less astronaut blood stem cells after the astronauts went to space. This would help address my research question of whether changes in astronaut stem cell levels could be impacting their white blood cell telomere length. Next, as I was writing up my research proposal, I made sure to include detailed steps about every process. Good research experiments are detailed, so as many factors as possible are accounted for. This helps you be more confident that any results you get are not from any unintended factors. So for my experiment, I made sure to list what quantities of materials I would use, how supplies would be stored, and exactly how tools from the Genes in Space toolkit would be used to analyze my experimental samples.
Awesome. So just to summarize, we want something that's specific, something that's fairly simple, and then a detailed use of that toolkit, right?
Yes.
Awesome. Well, thank you very much. Pristine, thanks for coming and sharing your experience and your feedback with everybody. Thanks to everybody who submitted their questions. We really hope that these answers will help you to develop some really robust and interesting creative proposals. Pristine, thanks again for coming.
Thanks for inviting me.